From Microbe TV, this is Office Hours for Wednesday, October 2nd, 2024. I'm Vincent Racaniello, and welcome to your viral corner of the internet where you can talk with a virologist and maybe get some questions answered, or like tonight, have a mini lecture. Let me welcome everyone. Good to see you all. And also our moderators. Who's here tonight? Andrew from New Zealand. Les is here from California. Tom is here from the Oregon Coast Range. Thanks for being here and moderating. I don't know how many people we have. 21 likes. My live stream thing is not showing up the concurrent users, interestingly, but it doesn't matter. Hit the like button if you can do that. So what's on tap for tonight? I want to talk about the Missouri H5N1 a little bit, and I want to do a mini lecture on how we detect viruses, and in particular PCR versus infectivity versus antigen tests, how they work, and what's the story with SARS-CoV-2, okay? Because a lot of people ask about those, so we'll talk about that. And, of course, there's your questions as well. So where are you from, folks? I am here in New Jersey. We're at 17C. And Philip is in Wales. Welcome back. Wayne is in Urbana, Illinois. Lise is in Columbus, Buenos Aires. Dagfinn is in Norway, 5C. Wow, it is chilly there. Yeah, well, it's going to be cold. It's Norway, right? Hello, Andrew from New Zealand. And Rima won't be here. Thank you for telling us. Frank is from Santa Barbara. 84. Kind of would like to still have 84 weather, but we don't here. That's the way it is. Visto Tutti is from Sydney, 19C and cloudy. Costella is in Ohio. Hello, Costella. Kang is in Chicago, 21C, clear skies. Tom is in the Oregon Coast Range, doing his restoration. Dagfinn's light correction, +5, not minus. Mike and Lori are in Seattle, 61F. B. Clark, Vancouver, 16C. John, 75F in Minneapolis. By the way, Tom says TWIV 1152 Origins paper. Next Sunday, or this Sunday, TWIV 1150. I always forget. 5. We discussed it on TWIV also, a deep dive on TWIV. So you get two different perspectives, but they're both the same. It's a very interesting and very important paper. More evidence for natural origin. Hard to really argue with it. Les says that Marburg is in Rwanda. That's not good. Nasty virus. Michael's in Mexico City, 14C, with a new woman president. Good for you. That's great. Yes. Okay. Who else is here? Environmental is in Allegheny County, upstate New York. Yep. Claire's from the UK. Environmental wants to know if anyone got Novavax, so you could chime in. Elizabeth is in West Virginia. Cheryl is in Santa Rosa. Hot. 101F. Wow. That's hot. That's really hot. Nebby is in Central Florida where it's 88. Anybody else taking? Well, why don't my viewers show up? It's weird. Oh, there it is. 50 people are here. Hit the like button. Track more. So we had a question up here. I'll take right away. Let's see. It was from Christopher. Hello, Christopher. What's known about COVID XCC? So this is the latest variant that has spread in Europe and now is spreading in the U.S. It's taking over. It's about 5%. So here I have some information. I wanted to talk about one aspect of it. So this is an NBC News article where what to know about XCC. Okay. So, 5.7% of the new cases in the past 2 weeks. It's a recombinant of two other strains, KS1 and KP33. So 2 viruses infected a cell, exchanged genetic information, and out came XCC. We don't know if it's more transmissible, but I would argue hardly know for any variant because there's so many other variables that confound it. Nobody bothers to sort them out. And here's the thing. Since the variant has only gotten a strong foothold in a handful of countries, it's possible that the reason for its success is it's simply in the right place at the right time. Oh, who said that when Alpha first emerged? In fact, D614G, I said, right place, right time. It's the founder effect. It's not clear if the virus is more contagious or if its spread was driven by something else like an increase in summer travel. How many times have I told you guys the Alpha's increase was due to an increase in people going back to their activities together, right? And now finally some people are saying this after 3 years. No evidence that it caused more severe illness. There's no evidence that it's going to evade the vaccine. So just another variant in the COVID play, SARS-CoV-2 play. So that's the story there. Okay. XEC. All right. What else? Was there anything here else before I dive into H5N1? Okay, so here's the story. Let's go back to the screen share. You Google for H5N1 Missouri because you heard there were a couple of cases supposedly there. Look at the different headlines you get. So the CDC is very straightforward. H5N1 bird flu response. We're going to look at that in a minute. But look at this. CIDRAP, Missouri investigates more possible human-to-human H5N1. Possible cluster of human flu, bird flu. Cluster of 8 possible bird flu cases now reported. Well, as you'll see, there's no evidence that more than one of them is H5N1. If we go to the CDC, CDC continues to respond to the challenge, blah, 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 posed by H5N1 in cows. Missouri case update. The states only reported H5 case. Only one. There's one confirmed H5 case. Okay. And then some other healthcare workers got sick who were exposed to this case. One was negative for flu. The second, they're looking for antibodies. They don't have it yet. 4 additional healthcare workers developed respiratory symptoms. None of these have been confirmed to be H5 so far. To date, only one case has been detected in Missouri. No contacts tested positive. All right. Yet, if you look at the headlines, we got a possible cluster. We got 8 possible clusters. I mean, yeah, it is possible, but it's not. Why would you make it? Oh, you want people to read your news? I get it. You need to make money. Okay, so that's the issue. Let's wait and see. There has been no person-to-person transmission in the U.S. of H5N1. It's very rare globally. And I'd be surprised if this were it, but let them do the tests. What is it that the one guy said? Where was that? I had a pretty good... what was that? CIDRAP maybe? Let's see. Yeah, here we go. Results of serology testing on the positive case are still pending. To date, only one has been detected. I already told you that. So that's it. Okay. That's the story with H5N1. We don't know what's going on. Any questions? GSD, hello. And Sanitas 21 and foggy. Picking plaques again. So this is good. We're going to talk about plaque assays tonight. Where's everybody? I don't know. 71 now. They're coming. They come slowly. All right. Bobby White wants to know, why is flu mist nasal spray not approved for people over 49? I would think it hasn't been tested. Let's see. Why is flu mist not recommended over 49? Apparently works better in kids. Clinical trials found it was not effective in people that age. How about that? It's approved for people from 2 to 49 years of age. And so the clinical trial where they tested the vaccine, they found that it just wasn't effective in people over 49. So they're not going to recommend it. And that's the way it should be, right? Okay. I'm reading a lot of things here. Eastern Pennsylvania, welcome. Hi, Doc. Lab theory people mentioned something about HIV parts in COVID as proof. No. Yeah, early the pandemic, when the genome of SARS-CoV-2 was deciphered, preprint came out saying it had bits of HIV-1 in it, and it was wrong. It was just wrong. They didn't know how to use their computer. And so, no, there are no bits of COVID. That's an old HIV. That's an old, wrong preprint. It never got to be published because it was totally wrong. But this is an example of how it's a preprint. It's up and it sticks, and so people will grab it and use it for their cases. Yeah. John says Columbia got a gift of $400 million for biomedical research. Let's look it up. Because yeah, the Roy and Diana Vagelos, it was expected. They're giving a lot of money to Columbia. Roy Vagelos got his MD there. Roy Vagelos was the CEO of Merck for many years. Now he's with Regeneron, has a lot of money. He has given much money over the years. In fact, there's a brand new education building on Fort Washington Avenue. It's beautiful, beautiful architecture. Big 13-story building, I think, with lots of glass. It's really good. And how much did he do? Almost $1 billion he has given. So that was August. Let's see what this one was earmarked for. Secure Columbia's leadership in biomedical science research and education. Single largest gift ever made to the medical school. It will expand the mission of the institute created last year to do research in basic science departments. And they're going to make a new biomedical research building at 167th and Audubon. That's up at Audubon Avenue. Where the hell is Audubon? Haven, Fort Washington, Broadway. It's east of Broadway? Oh, that's interesting. So they're going to build a new building with 55,000 square feet of lab space. Now, I haven't heard from my colleagues any new opportunities, but I presume it takes time for money to be distributed. They'll probably have some announced program, and then the building, of course, is going to go up, and they can fill it with new people who can presumably get NIH grants. That's how it works in universities. They build a building, then fill it with people who will presumably get grants from NIH, and those grants will let them do research, and it provides indirect costs to pay for the building. That's how it works. Fisto, what would happen if a person with human flu drinks raw milk with H5N1? Well, if they already have flu, well, if they drank it, they could be infected, because we know that if you feed mice milk, cow's milk that contains H5N1 virus, the mice will get infected. So I presume the human would get infected. And maybe you would get reassortment and new viruses made in that person. But we don't know, right? I don't know if there are people who drink H5N1 infected milk. I think it's all not sold, right? So it's not likely to happen. Yep. The exact route of infection for polio. Fecal oral contamination, right? So somebody is pooping out polio, POP, pooping out polio, and they get a little fecal contamination on their hands, which happens, right? And they don't wash their hands, maybe, and then they shake someone else's hands, and that person gets it on their hands, and then they touch their mouth. And kids do this a lot. Kids do it a lot, yeah. And so kids get polio often. But as you saw, there was an adult in Rockland County in summer of 2022 who got polio too. Didn't wash his hands. All right, so you eat the virus, it goes into your intestine, it reproduces, and then it enters the bloodstream. And my hypothesis was it goes to the muscles, and the virus enters the neuromuscular junction and then gets transported by retrograde axonal transport. It goes up the nerve to the brain, first to the spinal cord, right? So if your leg muscle gets poliovirus from the blood, the leg muscle is innervated, and that nerve goes to the spinal cord. It would be a lumbar level of the spinal cord because it's your lower limb. And then the virus would reproduce in neurons there, and it could, if it destroys enough neurons, the leg gets paralyzed. That's how it works. And in BS, you should think it involves retrograde transport in the axon. Yes. Now, we did an experiment in mice to show that if you inject poliovirus into the leg of a mouse, the leg where you injected it is the first one to be paralyzed, because the virus goes from that leg up to the cord, and those neurons that innervate that leg, the virus destroys and paralyzes. But if you cut the sciatic nerve before you inoculate poliovirus into the leg muscle, of course, then the leg muscle becomes paralyzed, right? So you cut the nerve, but the virus never makes its way into the spinal cord. You can then cut the sciatic nerve, you can inject poliovirus into the leg muscle, and then you can look in the spinal cord for a virus. And if you don't cut the sciatic nerve, you find virus there. If you cut the sciatic nerve, there's no virus there. So there you go. That's how it works. It's like it's some things that I know. Oh, boy. Good job. Oh, this is very nice. I always like to be here. When I see Dr. Vincent, I always feel safer. Like when COVID was ripping through everywhere, Twiv got me through a lot of stress. Well, I appreciate that. I appreciate that. I'm happy to help. As I say in this saying above, if you do your best, sometimes you'll resonate with someone. I guess we resonated. Neil said, I actually meant to ask, am I still protected after 4 vaccines? Protected from what? Not from infection. Nobody is. It is a myth that you're protected from infection. But from severe disease, it depends how old you are. If you have other comorbidities. If you're less than 65 and you're healthy, yeah, you're going to be protected against severe disease. no matter what variant is around. But, if you're older, all bets are off, no matter how many vaccines you get. That's why you have to take Paxlovid if you test positive and you're over 65 to prevent yourself from getting low O2 sat and going in the hospital. I just realized I have a hood on my jacket here. Yeah, but I can't use it because the wire's in the way. Yeah. But if I were chilly, I could put my hood up, but I'm not chilly. I get the ear wires in the back, which prevent me from pulling the hood up. I tried to look spooky. Maybe on Halloween I'll do it. Sean says, can you explain to us the reasoning behind your prediction? And Daniels, well, this is a pretty obscure question, John. Well, I happen to know what you're talking about. So if you look at the wastewater surveillance for SARS-CoV-2, let's look it up. We can go to Daniel's TWIV. Here, well, I'll show you what I'm doing here so you can watch me. So we'll go to more TWIV episodes here. And we will find clinical update. And let's find the where is this, the wastewater? Maybe I should search for water. Wastewater scan dashboard. There it is. Well, that's not it. No, that's not what I want. I want the thing that he does. Well, I'll get up his the show notes, which nobody ever sees, and you will get to see them shared with me. TWIV clinical update. Where is it? Here we go. Clinical update 1154. Okay. There you go. This is, I know it's small. I'll pop it up here. So this is SARS-CoV-2 by PCR in wastewater. And you can see September 23, about a year ago. It was high activity, but then it really went up in December, big peak in December, January. Then went down. And then this summer we had a peak, July, which we didn't have last summer. And now it's going down. So what I said is that it's going to stay down. It's not going up again. We're not going to have a winter peak because we already had this peak in late summer, fall. Now, this is just speculative and wishful thinking, actually. But Daniel says we're going to have a peak in a month or two. And he may be right. I may be right, but that's the thing about prediction. And especially with SARS-CoV-2, where no one really knows the seasonality yet. Is it going to be a winter disease or can't decide? Nobody knows. And if someone tells you they know, don't listen to them because they're wrong. They're just posturing. Okay, that's my reasoning, which is not really reasoning, but here, environmental. I live rurally, and at the farmer dinner that I went to last Wednesday, I met the guy who pioneered raw milk. And this was years and years and years ago before it was political. Wow. That's cool. And personally, I don't think anyone should be drinking cow's milk. I don't think we need cow's milk. Right? Because cave people didn't have cows, right? The early Homo sapiens didn't have cows. Well, I think cow's milk is a mechanism by the dairy industry to make money. It's big business. But you don't really need it. You can get calcium from other things. If you like it, that's fine. I don't particularly like milk. It's just I have some in cereal. That's about it. I would never drink a glass of milk. I just don't like it. Okay. Anything else? Okay, here we go. What's the effectiveness of updated flu mist versus injectable? Experts seem to be all over on this issue, but I would like to see actual data. That's interesting. So last night I did a live stream with a TWIV recording at a meeting in Brisbane, Australia. Did it remotely, right? And I asked this question of one of our hosts. So Kathy and Steph Lango were down there, and one lady, Jenna, who works on antibody responses to influenza viruses. She said that actually, yeah, she traditionally thought that flu mist wasn't any better than the injectable, but she said, particularly in young kids, it is better. I don't know what the age is, but she seemed to know that there were studies. I haven't found them. I don't have them at my hand here, but I could ask her to send them. And she said it's better than the inactivated for certain age groups, but obviously over 49 it doesn't work at all. So you don't want it, Lise. Okay, doesn't work. Hey, look, microbe meditations. I like that name. Where you from? Welcome. If you're over 65, won't COVID vaccines assure your immunological system is ramped up? For a short period. I have a paper here that we're going to do says mRNA vaccines don't give you any immune memory, no B cell memory. A little surprising to me. No memory B cells in anybody that they looked at. So I don't know. But if there is a boost, yeah, you get a boost for a couple of months. Get a bunch of antibodies made, which will protect you, but you don't get any long-term protection as far as I can tell. So you have two choices. You could get a booster all the time, whenever they recommend it, or you could just sit with what you have, and if you get test positive or you're over 65, you take Paxlovid. I know you can't do that in every country. So instead, booster. And Margot said, I keep thinking of Amy's 5 years in 2020. Yeah, it'll be 5 years next year. The pandemic is over. Well, actually, Amy said to me the other day, the pandemic is over. So it's almost 5 years, right? 20, 21, 22, 23, 24. Yeah, it is 5 years. So that's good. Should anyone be taking PCR tests for COVID anymore? No. I'll show you why today. The mini lecture is actually going to address that, John. It's a very good question. This morning I got up. I said, what am I going to talk to them about today? And I said, I want to talk about detecting viruses with a goal of understanding how PCR, rapid antigen infectivity tests work. Yeah. Yes. Hello, Sandy. I didn't forget it, Sandy. Kang says, what would make a virus more infectious? Well, so let's say you had a virus where you need 100 infectious particles to initiate an infection in your nose. And if you could make changes to the virus, or if the virus underwent changes, because nobody can make those changes, nobody knows what to do. If the virus underwent changes with that, now it would only require 10 PFU. That would be more infectious, right? Now, what kinds of changes would do that? That's a good question. What kinds of changes would do that? Make the particle more stable. Make the proteins more stable. Maybe put more proteins in the particle. I'm just waving my hands here because I don't know what would make it more infectious. Maybe give it an immune antagonist. This can happen by evolution, by viral evolution. The immune antagonists can get better. But yeah, the effect is that you need less virus to infect the host. Hit the like button. We have 113 people here, 66 likes. So hit that like button, folks. Hello, Tracy from Chapel Hill, where it's 20C. Bobby says a Marburg outbreak in Rwanda. NPR article says Marburg is an especially scary virus. How scared should we be of its outbreak? Well, this is clickbait because the West Africa Ebola outbreak had over 20,000 cases and it barely spread out of those countries. It just doesn't spread very well. And Marburg is no different. So I think you'd be concerned if you're in Rwanda. And I suspect that the Ebola vaccine will cross-protect, but I'm not sure. I'd have to look it up. Oh, Microbe is in Indianapolis. Thank you very much. Welcome. I've been here before. I like the name, though. It's good. It's really good. Well, I love you all. I love all of you. Racaniello, there's no end. It's okay, though. Love you all. Right? I can't do this very well. I think if you do it with these, yeah, this is better. Okay, love you all for coming here and partaking of this community. It's great. Rodmir asks do we know if viral load matters for infection or disease severity for COVID or flu? We don't know. I mean, for COVID, I mean, there are challenge experiments with flu that have been done. I don't know if they varied the dose, though. And the same with COVID. You know, the one challenge experiment was done, and they only used one dose. Well, by the way, so anyway, I suspect if you got more virus in you, it would accelerate disease. I don't think it would necessarily alter disease severity unless it overwhelmed innate immunity, and that could lead to more severe disease because you might get an overreaction of the adaptive response and a lot of inflammation and so forth. So it could be. But, you know, people who say the viral load does matter for these viruses, it's not correct because the experiments haven't been done. Okay. What else did I say? Okay, let's do a mini lecture, and I'll come back to questions. I want to talk about measuring viruses. So I got to start the play here. Play. Measuring viruses. I want to talk about methods of detecting viruses so that you can understand an infectivity assay, a COVID PCR test, and a COVID antigen test, okay? So measuring viruses, it's all about measuring viruses which are made in infected cells, right? So this is a diagram of a cell and a virus getting into that cell and making more viruses. This is called the infectious cycle. And we divide this into steps to study it, but of course it's a continuum in reality. But the point here is that when you're infected with virus particles, the viruses are taken up into individual cells wherever the portal of entry is, whether it's your upper tract or your gastrointestinal tract. And then those viruses reproduce in those cells and make lots of viruses that come out and go elsewhere to other cells in you and to other people maybe. So the key is, how do we measure them? How do we measure viruses so that we can make intelligent conclusions about the properties of the virus? All right. So first of all, a key component is that we use cells that are cultured in the laboratory. They don't go to the opera. No, not that kind of culture. We grow them in plastic dishes, and the cells grow nicely in sheets. And this slide just illustrates 3 different kinds of cell culture, where you can make these and use them to grow viruses. And that's part of the assay of viruses I'm going to talk about. So we have primary cells, which are made from a tissue like foreskin, fibroblasts. People in hospitals throw out foreskins every day. So you can go get them and make nice cells from them, and they will grow nicely on a plastic dish, and viruses can grow in them. We have cell lines which grow forever. Your mouse cell lines, HeLa cell lines, many others. So you don't have to keep making them. You don't have to go get a foreskin every week or two and make cells out of it. You just use the cell. But these are abnormal. They're transformed. They grow forever. They're immortal, and they're not normal by any means. Anyway, these cells are the key to growing viruses nowadays. And when you infect cells, the cells can die sometimes. Like here's an example of cells infected with poliovirus. And on the top left, we have × 0. We just put the virus here. You can see these cells look lovely, right? Just lovely. 4 hours later, some of the cells are getting round. That's because the poliovirus reproduces really rapidly and causes changes in the cell. So they get round and then they detach from the plastic. And then I think by 12 hours, they're all lysed. So many viruses do this. During the infection of cells, they can cause damage. But the key here is that we can grow viruses in cells, and that's part of our ability to assay viruses. Okay? So here's the fundamental question for today. Well, for me, it is fundamental. It's for a diagnostic, it's not fundamental, as you will see. But I want to know how many viruses are in a sample. So there are two broad ways that we can do this. We can measure infectivity, or we can measure physical virus particles and their components. Right? So infectivity tells you how many infectious viruses there are there in the sample, and then you can't do that for all viruses. So in that case, you look at the physical particles and components. So this is a picture from my lab. These were Amy's viruses a couple of years ago. All these tubes have viruses in them, and she's doing plaque assays to measure how much virus is in them. All experiments, and here's some plaque assay plates here. So what is a plaque assay? This is the greatest assay ever developed. It is a measure of infectivity. First developed in the 1930s to study the multiplication of bacteriophages, viruses that infect bacteria. So what we have here is a petri dish, right? It's got agar, and it's covered with bacteria. This cloudy layer is a layer of bacteria that have grown on it. And these holes are clearings caused by viruses. They're plaques. They are plaques. The virus is killing the bacteria, so it's clearing the area. And so we can count them and say there's so many plaque-forming units in this culture, in this tube of virus. So that's the beauty here. The plaque is caused by a virus, often a single virus particle, and therefore it is a direct measure of infectiousness of the virus. It is not like PCR or antigen tests, which are not direct measures of infectiousness. There's no readout in a PCR for infectivity. It's just DNA. There's no readout in an antigen test for infectivity. It's just protein. This and a related assay, it's the only way you can measure infectivity. So this was first developed in the 30s for phages. And then in 1952, Renato Del Becco said, I need this assay for animal viruses because it's going to enable us to do a lot of things. So he published a paper here in the Proceedings of the National Academy of Sciences, April 29th, 1952. I wasn't born yet. Production of plaques in monolayer tissue cultures by single particles of an animal virus. And so here was this plaque assay up here on the upper left. So this revolutionized the study of animal viruses. You could quantify, and that's the key, quantify or quantitate the number of infectious particles. And then you can isolate the virus that caused a single plaque and have a genetically pure stock of the virus. Really important. So how do you do a plaque assay? This is not a diagnostic test, right? No, I mean, you could, in a specialized lab, if you suspected you had SARS-CoV-2 or more likely influenza virus, they could do a plaque assay, but they're not going to. They would do a rapid antigen test. But if you want to do a plaque assay, you take your tube of virus and you take 0.1 milliliters of the virus and you add it to 0.9 ml of saline. So it's a 10-fold dilution. You mix it up, you take out 0.1, you put it in another tube, and you keep doing that. So you get what we call serial 10-fold dilutions. And here you can go all the way up to 10 to the minus 7th, right? Dilution. There's great dilution there. All right, and then you take samples from each of these dilutions and you add them to cells in culture. You have dishes with cells in culture. You take off the liquid overlay, you add a 0.1 ml of virus. You let the virus attach for an hour, and then you add an agar overlay to these plates and incubate them. And after a few days, depending on the virus, you will get plaques. The virus will kill the cells, and you can stain the monolayers and nicely see these plaques. And then you can quantify. You can say how many plaque-forming units were in the original virus stock. Well, here we have 17 on this plate here. There's too many on this one and too few on this one. So this is good. This is the Goldilocks plate. So the titer is 17 times 1.7 times 10 to the 8th. The dilution is 10 to the 7th, 10 to the minus 6 plus another tenfold. So it would be 17 times 10 to the 7. You just move the decimal over, 1.7 times 10 to the 8th. Plaque-forming units per mil in that original virus stock. So that tells you how many virus particles are there. And if you wanted to infect an animal, then you could take a certain amount to give you a certain number of PFU. Precise. You know exactly what you're adding to the animal. All right, so how does this work? Here on the top is a blow up. You put dilute stocks of virus on these plates, and here a single cell is infected. It will die, and the cell is gone. But those viruses will spread to neighboring cells, so this hole in the monolayer gets bigger and bigger. And the key is that you have an agar overlay on top that restricts the movement of the virus in the cell culture. If you had a liquid overlay, the virus would just spread everywhere by diffusion and all the cells would be dead. You couldn't count any plaques, right? So the agar overlay is the key. And here at the bottom are some microscopic photos. Here on the lower left, this is a single plaque. You can see the cell monolayer is intact, and then within the plaque, the cells are rounded up and detached. And when you take off the agar overlay and you stain the monolayer, all these cells wash away, and then the living cells get stained purple if you're using like crystal violet. Now, some viruses don't form plaques. Here on the right is a herpes virus. Doesn't form a plaque, but we can put a gene in the virus that makes blue color, and we can see plaques basically the same way. These cells aren't dying, but they're infected, so they're making the blue dye. It's very cool. There's a movie of a plaque formation. So what they did here, let me stop it. Let's go to the beginning. Okay, they infected cells with vaccinia virus, and then after a few hours, they identified what looked like was going to be a plaque. You could see there were some detached cells here. There's a little hole. So they pointed a camera at it, and they took a frame every so many minutes. And this timer goes up to 15 hours. So this is time lapse. And you can see the plaques getting bigger and bigger and bigger. First thing you notice is that the cells get refractory. They're getting light colored, right? Because they're now refracting light back rather than passing it, because the virus is killing them, and that causes the refractory change. And of course, the cells are coming off the plate as well. And it's a very nice symmetric event, right? The infection is spreading outward from the point in the middle. So again, this is under an agar overlay, which restricts to diffusion, or maybe a semi-solid, but in any case, that restricts to diffusion. Greatest movie ever made, folks. No doubt about it. Now, some viruses do not form plaques. So what do you do? Well, you can still measure infectivity. You have to use what's called an endpoint dilution assay. So what you do is you take a multi-well plate, which is shown up here, and you make dilutions of your virus, just like before, -2, 3, 4, 5, 6, 7. And then you plate out 0.1 ml. And these wells are cells, little monolayers. They're just tiny compared to the ones we used before. You plate 0.1 ml of your virus dilution across the rows here. So every well in this row will get 0.1 ml of -2 dilution, and the same for the next, -3, -4. Okay? Then you let the virus absorb for an hour. You put medium on the cells. You put it in the incubator. And after X number of days, which you've figured out ahead of time, you take out the medium and you stain the cells and you ask which cells show cytopathic effect, which cells are killed by virus infection. And you score them here. So by CPE plus, all these cells were wiped out by the virus, and the next row all wiped out. And then the next one, -4, 1 was not wiped out. And then the next one, only 5 were wiped out. That's 50%. And then the ones beyond that are all wiped out. Excuse me. So this is the 50% endpoint. And that's what you want to do. Calculate the dilution at which half of the wells show CPE, cytopathic effect. And we call this the tissue culture infectious dose 50%. So there's a good way of quantifying infectivity if you don't have a plaque assay. And this is often done in industry. It's a very rapid assay. And you can now say, this is the TCID 50, and you can say my virus stock has, knowing the dilution, you could say my virus stock has X TCID 50 doses in it. And then when you do your experiment, you can use that to make sure you have the right amount of virus. Okay? So those are infectivity assays. One thing I have to tell you is that not every virus particle is infectious. For reasons that are complicated, for many viruses, most of the particles are not infectious. So here we call this the particle to PFU ratio. The number of physical particles divided by the number of infectious particles determined, say, by a plaque assay. So look at these ratios. Papillomavirus is 10,000. 1 in 10,000 viruses are infectious, will form a plaque. Poliovirus is between 30 and 1,000. What else here? Herpes, influenza virus, 20 to 50. And the king here is Semliki forest virus. Almost every particle is infectious. So you have to just keep this in mind. Cells make a lot of particles, but they're not always infectious. Not clear why. I think the infectious cycle is complicated. Lots of mistakes get made. You still end up with a lot of infectivity, but there's also a lot of non-infectious particles, and they may have their own functions. Okay, so now, what if you can't do an infectivity assay? Or if you want to do a diagnostic test, you're not going to do an infectivity assay. You want some physical measurement. Here's some assays that we use. Hemagglutination, we'll talk about that. You could take pictures of the viruses, but that's not very practical. You can measure enzymes in virus particles. Retroviruses have reverse transcriptase in them, so you can measure that. You can use antibodies. You can do ELISAs, serology, to measure either viral antibodies or viral proteins. We're going to talk about that, right? Rapid antigen test, RAT. Or you can do nucleic acid-based tests like PCR. So I want to go through some of these and compare them, explain what they are, and then talk about their use in the context of COVID, the use of PCR versus infectivity versus RAT, rapid antigen test. So hemagglutination test is done for viruses that bind red blood cells. So influenza virus is the classic example. It will bind red blood cells. It doesn't reproduce in red blood cells, but you can use this for a rapid assay. You buy chicken red blood cells and you wash them, you spin them out, wash them, and then you mix them with dilutions of influenza virus. And here are the dilutions of influenza virus at the top here. And then you just let the samples sit in these little 96 well plates. And if there's no virus present, the red blood cells will tumble to the bottom of the well, form a nice little button. Because the virus is actually binding the red blood cells, and because there's so many virus particles there, the red blood cells will attach to another red blood cell with a virus particle on it and form this lattice. So on the bottom here is a sample that's positive for influenza virus. And you can see many of the wells, there's no button. That's because the virus is sticking the red blood cells together. And also only beyond a certain dilution do you get the buttons, because now we've diluted the virus out and it can no longer agglutinate these red blood cells. So you would say here the HA hemagglutination titer is 1 to 512. It's the last dilution at which you got hemagglutination. 112 actually, the inverse. Okay, hemagglutination. You can do this really quickly. You can do it in less than an hour. Doesn't measure infectivity. but it tells you if you got virus particles there. Now let's talk about serological assays. These are typically called enzyme-linked immunosorbent assays, or ELISAs. We can use them to detect antigens or viral antigens or antibodies to viruses. And here are the two different assays here. On the left, we're looking for viral antigens. So this would be a rapid antigen test. You take an antibody, what's called a capture antibody, which is to the protein you want to measure. So for a rapid antigen test for COVID, it would be the nucleoprotein. You stick it on a solid support, and then you put your sample over, a nasal wash, for example. And if there's viral antigen present, it will bind the antibody. And then how do you detect it? Use a second antibody that will bind your antigen. So both of these bind your antigen. One is stuck to the solid support, and one is got an indicator that you can read out. And it would be a line on a test, which I'll show you in a moment. So that's looking for viral antigens. You can also look for antibodies, in which case you stick the antigen on the plastic surface, and now you wash over serum, and if there's antibodies to the antigen in your serum, they will bind, and then you detect those antiviral antigens with a second anti-IgG antibody that has some kind of indicator that you can read out, that you can measure, quantify very, very easily. Right, so those are ELISA tests. And the one on the left is the basis for the rapid antigen tests for many viruses, and of course for COVID. Now, you can use antibodies to detect infected cells. This is an experiment from the very first paper to describe SARS-CoV-2 in early 2020. They had taken bronchoalveolar wash from some of the patients. They put them on cells, and the cells got infected. And you can see here on the left, these are the cells, and on the right, they've added an antibody to nucleocapsid protein, and they're fluorescing red because these cells are virus infected and they contain nucleocapsid protein. So again, if you want to know if a cell is infected, you can throw an antibody on it, and the antibody will make a signal if the cell has viral protein in it. But here's the format you're familiar with for these rapid antigen tests, right? It's a lateral flow assay. It comes in a plastic cartridge, and you see a little window where you're going to see these lines, a test line and a positive line. And so I'm going to tell you what the basis for those lines are. So this is what's in that plastic container. Take the plastic container off. You've got an absorbent piece of paper here, thick paper, and then you have different regions with different reagents. So at the very end is where you put your sample. So you do a nasal wash, you put it here, and then it begins to move down the filter paper by capillary flow. So the fluid will move down and carry whatever is in it past all these regions. So the first region here are pre-adsorbed to the pad, monoclonal antibodies to your antigens that are linked to colloidal gold. Those little yellow circles are colloidal gold particles. So if your sample has SARS-CoV-2 N protein in it, as it moves past this first region, it's going to be bound by these antibodies. And so that's shown on the second slide here. We have the antibodies now are moving up the pad. They have captured the N protein here, and they still have their colloidal gold, and they're moving up. And then they reach the first line, which is full of antibodies that have been stuck to it. And these are antibodies to N protein. So if any of these antibodies have captured N protein will bind to these antibodies, and they're present in a line, and the colloidal gold will then make that nice black line for you. It can be other things as well, but colloidal gold is one thing that'll give you a black line if you're antigen positive and they all line up in this manner. And then finally, the last line is a control line to make sure your test is valid. It has antibodies to antibodies. Okay? And so whether or not the antibodies, some of the antibodies are going to keep flowing past the test line, and they will bind this control line. And they have colloidal gold on them, so you'll get a line, and that tells you the test has worked. Now, if you're negative and don't have SARS-CoV-2, you will have no test line, of course, but you will have a control line to show that the assay worked. If there's no control line, you throw it out. You have to do it again because something didn't work. Okay, so that's the basis for a rapid antigen test. You can, develop these typically in less than an hour, and it's all depending on antibody-antigen interactions. Really beautiful. Okay, so what about nucleic acids? Right? We use sequencing all the time to sequence genomes. We use what's called deep sequencing, where we can do very, very large numbers of bases in a single day, and we have millions of SARS-CoV-2 genomes as a consequence of this. We don't use this for diagnosis, but this is one way that you can detect nucleic acids in a sample. And just for your information, this technology has evolved amazingly. 10 years ago, or more than 10 years ago now, the first human genome was sequenced at a cost of $3 billion. Today, you can do it in a day for less than $1,000. It's just bloody amazing. Bloody amazing. When you get genome sequences, then you can compare viruses. And you can say, what's related to what? You build what's called a phylogenetic tree. And you use a computer program to pairwise compare. Let's say you have 10 virus sequences from an outbreak. You want to know how they're related to each other. Are they the same? Is one more related to another? So the computer will do pairwise comparisons and then build a tree with the best pairwise comparison. So you can see here virus 1 and 2 are the most closely related, 3, 4, 5 not so much, with 6, 7, these are all on their own here, 8, and then 9 and 10. And then these would have common ancestors. This green box would be a common ancestor of 1 and 2. And then there would be an ancestor that shares among 1 through 4, et cetera, all the way back to this blue square, which is called the root, which is the presumed ancestor of everyone. And this line here is a scale. It means the degree of genetic change. So it's changes per length of sequence. So you take sequences and you can do this and ask who's related to what. And this is what was done early in the COVID pandemic. We looked at, in red here, the genome sequences of some of the first isolates of SARS-CoV-2 from Wuhan. You can see they all clustered, right? They're all highly related to each other. And then the most closely related virus was a sequence from a bat isolate called RATG13, the famous or infamous RATG13, which is about 1,200 nucleotides different. So it was the closest at the time, but it is not an ancestor. You can see it groups with these SARS-CoV-2 isolates. It's not an ancestor. If it were an ancestor, it would be back here. And then a bunch of bat SARS-related coronaviruses here. And then there's SARS-1 here as well. Here, bat's bat. Where's the human SARS-1? Here's MERS coronavirus, a lot of the human common cold coronaviruses you can see here. Here we go, SARS 2001 up here. Very distantly related. Well, not distant, it's about 80% amino acid nucleotide identity. So again, these new viruses were not SARS-1. You could tell right away from this. And we didn't have an ancestor for SARS-CoV-2. Anyway, that's why sequencing and phylogenetic trees are useful. And then finally, that brings us to PCR, our last assay, for components of viruses, right? Nucleic acids, not an infectivity assay. Just like antigen is measuring protein. It's not measuring infectious virus like a Plaque assay does. With polymerase chain reaction, you take a small amount of nucleic acid. It can be DNA or RNA. If it's RNA, you first convert it to DNA using reverse transcriptase. And then you have double-stranded DNA. And then you add a few primers that are going to bind to each strand of DNA and make a product. And what you do is you take your DNA sample, you heat it 100 degrees, you denature the strands, you add your primers, and then there's an enzyme in there, DNA polymerase, that will make both of these double-stranded. So now you've gone from one piece of DNA to two. And you repeat that, you denature, you add primers, you extend. Now you have 4. So you're doing exponential growth. So you can have a very small amount of nucleic acid and pick it up right away. And the number of cycles you need to detect the product is called the cycle threshold. The more nucleic acid you have in a specimen, say the more viral nucleic acid, the lower number of cycles you need to detect the product. So this was used early in the pandemic to detect infections. It is not used much anymore because we have rapid antigen tests and PCR is too sensitive, because we got into a situation where people would test positive by PCR for days and days and days. They were not infectious, but you're positive, you got to stay home, that kind of thing. And here's the issue. PCR product is not the same as infectious virus. This is an experiment done with Zika virus, actually, before the pandemic. Now here you infect mice with Zika virus, male mice, and they shed virus in seminal fluids. And you can take seminal fluids from the mice and measure infectivity. That's shown in red by a Plaque assay. And you can see that you can't find any more infectious virus after about 21 days or so. However, if you do PCR on the seminal fluid, you can detect Zika RNA way beyond 20 days, all the way out to 60 days. You still get a signal. But there's no infectivity there. So should we, if this were SARS-CoV-2, should you stay home because you're PCR positive? Of course not. You have to figure out the right thing to do to help society, which unfortunately we didn't do because a lot of people could not put their head around this idea that PCR product is not infectious virus. So, and if you've vasectomized the mice, they don't shed infectious virus, but they still shed viral RNA. And what is this RNA? It's bits of the viral genome that stick around. They're broken. They're not infectious. This happens with every RNA virus. It happens with SARS-CoV-2. Here's an experiment that was done in people by Plaque assay and PCR. So you can do a Plaque assay with SARS-CoV-2 that's shown there on the right. And you take people who have been infected for so many days. You can see day 2 through 22. You take a sample from them. There's a pharyngeal sample. You do a Plaque assay. You get a peak of virus around day 6. And then after day 12, you don't get any more infectious virus. Yet if you do PCR, you get PCR positivity long after viral infectivity is gone. And so this is the conundrum. What do we do? And we never reached a consensus. Now we have antigen tests, and we can use that to guide us. So let's move away from PCR. Let's talk about the sensitivity of antigen tests and what they do and do not tell you. So I found this paper, which was a couple of years old, Usefulness of Antigen Testing in Predicting Contagiousness in COVID-19. Now, first off the bat, contagious is the wrong word because we don't know. Contagion means movement from one person to another, right? We don't know how many infectious particles you need. One may not be enough. If you're shedding 1 particle per milliliter in your nasopharyngeal fluid, that may not be enough to infect someone. Maybe you need 100. So if you had 10, you could go to work, right? But we have never figured out how many particles people shed and how many are needed to infect another person. So this is wrong. They're not predicting contagiousness, but what they're doing is measuring when you have infectious virus. That's more correct in it. So here's the experiment. They took a bunch of samples from people, and they did PCR, they did an antigen test, and they did viral culture. And these samples are from 1 to 5 days after symptom onset. That's the light-colored bars. And then we have gray, 6 to 11 days after symptom onset. And then we have more than 11 days after symptom onset. And then we have some asymptomatic people. And then they do a RT-PCR, an antigen test, a viral culture. And then these are the percent of the samples in this test that were positive. So the most sensitive is PCR, right? 70% with at days 1 to 5. It goes up 6 to 11 and greater than 11. And the asymptomatic people have less nucleic acid in them. The antigen test is about half as good at 1 to 5 days and 6 to 11 days. More than 11 days in this study didn't pick up any positivity. And then some of the asymptomatic people were positive. Now, the viral culture is kind of comparable to the antigen test in terms of percent positivity, you can see here. But this is qualitative. It is not quantitative. It is not a plaque assay. Hence, it cannot inform contagiousness. So what they do here is they take a sample, the same sample that was used for PCR or the antigen test. They divide it into 3, and they put a third of it on cells in culture. They just throw it on a monolith. They don't do a plaque assay. They just say, does virus grow out? So there could be 1 PFU, there could be 10, there could be 1,000,000 in that sample. You would never know because they don't do a plaque assay. It's unfortunate. This is constant practice that I've seen in the last few years. People don't do the plaque assay. And that's why this is not useful, because all it's telling you is that there's a virus there. So if you have a positive antigen test, it's about 50%, 40% sensitivity if you're infected. It pretty much correlates with viral culture. So yeah, there's virus there. If you get a positive antigen test, at least at these days, up to day 11, there's virus present. But you have no clue how much virus. You may be shedding very little. It would still turn up positive in an antigen test and a viral culture test, and you would not need to be quarantined. So this is why the CDC has been playing with, what to do with these antigen tests, and they pair it with symptoms as well. Now, the good news is a negative antigen test is 99% predictive of no infectious virus. Okay, so you can see here over 11 days, the antigen test agreed with viral culture. There's no virus, there's no infectious virus in that, and there's no antigen, but you do have to repeat it just to be sure. So that's the value of an antigen test. This is why we use antigen tests now, because they can tell you, do I have virus? And if you have any virus at all, that's considered not good, which is not fair. I don't think you should have to stay home if you have very small amounts of virus that would not infect someone. We don't even know what that is. And that's a failing of the system, I think. Okay, that's that. Hope you find that useful, and I hope you're going to ask me some questions now about this. I hope it resonated with you. And there you go. Viral load. Yeah, we don't know the viral loads. We just know, is it culture positive? I don't care if it's culture positive. I want to know how many infectious particles there are. Tom saw a talk by Linda Zhang. They gave people with long COVID the Paxlovid. It didn't help their participants. Okay, well, that's unfortunate. Sorry to hear that. I think you speculated before that more efficient viral packaging could result in better infectivity. Yeah, so remember the particle to PFU ratio? If you could somehow reduce that from a few hundred to 10, that would make you more infectious. And one way you could do that was to improve genome packaging, which I think is a big defect. But there are other mutations are another way too. Lower the mutation rate. Hello, Yu Xing from Hangzhou. Welcome back. Hello, Will. Welcome from... where are you from? China. I can't remember where. Sorry. Hangzhou is from... Yu Xing is from Hangzhou. Are you from Hangzhou? I don't remember. I'm sorry. John says, I don't think I've understood the auger overlay until this moment. And it's the 4th time I've seen it explained. Yeah, sometimes it takes multiple times. I've often had to read things a few times to get it. It's fine. It's totally fine. Okay, I7 of 7 asks, in a plaque assay, how do you ensure the diluted virus stock is evenly placed so you don't get a pool of virus? Oh, so you pipette 0.1 ml on, and then you shake the plate, then you turn it 90 degrees and shake it, turn it and shake it, turn it and shake it 4 times. And that gives you great distribution. Of course, your incubator has to be level too, right? But you can do that. GSD just mailed a double layer plate I recently made for phage stock titering to the incubator. Oh, cool. To the email address. Very good. A picture of it. I'll have a look. Thank you. What stops the exponential growth of vaccinia virus? Well, if you let the plaque assay go, it would keep getting bigger and bigger until all the plaques merged. And all the cells would be dead. And that would be the end of it because the cells would be died. There would be no more for infection. But you typically stop the plaque assay when the plaques are big enough to see visually, and you can count them, right? But in a cell system where you're doing infection of cells with no immune system, what stops the growth is no more cells. It is more complicated in a person, right? Because you're not going to run out of cells for a long time. Yeah, it's going to be the immune system that's going to stop the growth. Exactly. Good questions. John, the colony forming unit I'm hearing about on TWIM sounds like the bacteria equivalent of PABO. Yeah, more or less. You take bacteria, you make suspensions, you dilute them, and you plate them on agar, and they're going to form colonies, not plaques. So the individual bacteria will grow, they'll multiply and form individual colonies, and we call those colony forming units, right? Same idea. Nanoparticle tracking analysis. You mean to count plaques? I mean, yes, you could do that. There are machines that count plaques for you, for sure, but maybe you mean something else. I don't know. Just occurred to me that a bunch of our regular virophiles joined from the southeast U.S. I hope everyone is okay. Is everybody okay? Let us know. How can I control the virus stock? What do you mean by control? I don't know what you mean. Can you restate it? You mean keep it secure or what? I'm not sure what you mean. I hope someone from CDC is watching. Unlikely. They will say, I'm too busy. I can't watch this live stream stuff. Social media crap. Well, it's education, folks. It's a new way of educating. Daniel, no worries. I'm glad you're here. If you missed the mini lecture, you should go back and listen. It's really, I think it's useful. Martha is in Charleston, South Carolina. Are you okay? I know that Michael Schmidt is okay. He was in Charleston too. Yeah, he's okay. So I guess you're okay, Martha. Bernard is in Chicago, 60s and delightful. A urine test I saw was positive when the control line was present, but no line for the substance. Well, if the control line is positive and the test line is negative, that means whatever you're looking for is not present, if I understand you correctly. Right? If the test line is positive and the control line is negative, you can't trust it. Something may have happened with the antibodies to bind aberrantly at the test line, so you have to redo it. This is exactly what I used to teach new PhD students. Population and evolutionary genetics. Yeah, the most fundamental stuff is often the best. Why not keep going back to it? This is pure gold of knowledge telling. Thank you so much. I've told the story many, many times. Yeah. We have some new moderators. We have Steph SF. Welcome, Steph, and Barb Mac UK. Welcome. Cool. Joining. Thanks for joining. I know this is silly, but I think one of the best features is that I can see Vincent's cursor. Yeah, I figured out how to make it work on this Livestream software, because in my lectures, you don't see a cursor because I use a pointer in the classroom and I'm capturing the lecture on my laptop with a screen capture. So I'm not using the laptop to do the cursor. Now you may say, Vincent, why don't you just stand behind the laptop and use the cursor and then it'll get recorded. I can't stand behind my laptop. I have to walk around the room. I wander back and forth and up and down to make myself feel that I'm right there, right? I want to stand. You don't stand behind a table or a lectern when you lecture. That's, you're hiding from the students. You want to mingle with them. So that's why. Unfortunately, yeah, I could put a camera up and capture the screen, but it wouldn't be as good. It would be more complicated. And so here's what you get. So you come to the live stream and get the mini lecture version with the cursor. I think it makes sense. Yeah. Rapid test gives insight into how the intensity of the line's color can vary. Right. If there are more antibodies binding your antigen, you will get more intense color, because if it's colloidal gold, for example, there are going to be more gold particles lining up and you're going to get darker color. What that means, though, it's another story. I'm not sure what it means, because there's, the rapid antigen test agrees with viral culture positivity, but we don't know how many viruses are in that. So I would love to do a study where instead of just doing viral culture positivity, they quantified the virus by Plaque assay and see if that made a difference with the NP rapid antigen test. Yeah. Hello, Vanity Nutrition is here also. Oh, welcome. Hey, welcome. I know Professor Vincent who was new to science 5 years ago. I asked some stupid questions. No, they say it's not stupid to ask, but I learned so much. There are no stupid questions. I want you to ask your questions so that you can learn. And there are some professors who will say that's a stupid question, but I would never say that, because I want you to learn. So I want you to feel that you can ask anything. So every question goes. Yeah. Someone collects mouse semen. Yeah. I think they did dissect it out. They euthanize the mice, right? And they dissect out the gland so that they get the semen. Yeah. What would be a good visual analog for visualizing germ particles for hand washing? They have this dye that you can put on your hands, right, which simulates bacteria. It's not bacteria, but it's a dye. So you put it on your hands and you put it under a light and it glows, and you can see how much of the dye you have on your hands. And then you go wash your hands and you put them under the light and you see how crappy a job you did at washing your hands, because invariably most people don't wash their hands very well, and there's a lot of glowing dye still on their hands. That's a very good thing to do. It's not, it's a measure of hand washing. It's not germ particles, but that's what came to mind. Yep. Are there methods of creating flu vaccine eggs, dog cells, and insects? Not sure I understand the dogs. You can make dog cells from dogs, yeah. You can make insect cells from insects, yes, for sure. But eggs, yeah, you just get an embryonated chicken egg and you can grow influenza viruses in those. And that's how many flu vaccines are grown in this country, yeah. Okay, so do people with egg allergies actually get reactions from egg-derived vaccines? So I looked, I read a paper years ago which did this study, and there's no, the amount of egg antigen in the flu vaccine is so low, it does not cause egg allergies. And this study is if you search for my virology blog, this topic, you'll find it. I wrote about it on the blog years ago. However, they still recommend if you have egg allergies not to get the egg vaccine, just in case, right? Because it was a limited study, and there's going to be someone somewhere that's very reactive to the small amount of egg protein that's present in the vaccine. So you might as well just be safe, right? That's the story, for sure. Yep. Do they do plaque assays? Very few people did. I showed you that one slide that had a plaque assay of SARS-CoV-2. That's that experiment where they compared PCR versus infectivity by plaque assay. They had to do a plaque assay. But most people do not use plaque assays. They do PCR or antigen test. And if they're going to do infectivity, they just do viral culture. Right? In a plaque assay, you make dilutions of your virus and you plate them on plates and you cover them with agar so you can count plaques. A viral culture, you don't do any dilutions. You just take the nasopharyngeal wash, you throw it on the cells, you put a liquid overlay on top, and you ask, does the virus grow? So there could have been 1 PFU, 10, 100, 1 million. You have no idea. It's not quantitative, and in my opinion, it's not useful. Unless you want to say, if you have any virus particles at all, you got to stay home. And that's with your antigen test, it tracks with the presence of infectious virus, right? So it's not necessarily infectious virus that is causing the test, right? It's a protein. But it correlates with there being infectious virus in the sample. And so even if there's 1 PFU per mil, you're going to tell people you need to stay home, which I think isn't right, but we've never moved beyond that. We never moved beyond that. Now I know some acronyms and why you love plaque assays so much. I think the plaque assay is a fabulous assay, yeah. And yes, you can always learn some acronyms. Yep. How sticky are viruses? Oh, if you had some viruses on your finger, you'd never be able to flick it. No, they're pretty electrostatic, right? You'd have to wash them pretty hard with soap and water to get them off. Yeah. I mean, if you had a lot of fluid on your fingers, you could flick them, and some of the fluid-containing virus would go off. But if you got it to the point where you rubbed the fluid into your skin, no, you wouldn't be able to flick it. No. Nope. It's interesting. Hello, Mark Fuccio. Enjoy your grill. That's great. How is infectious dose determined? I don't know what you mean. Infectious dose for, in other words, well, let's put it this way. I'm going to make an assumption about what you're asking, and you can correct me. Let's say you want to do an animal experiment. You want to infect animals and treat them with an antiviral. So you have to make sure, and you're going to use 10 animals, and you have to give them all the same amount of virus, right? So you can say, I'm going to give you 10 PFU each or 10 TCID50s. But you have to first ask, how much virus will infect all the animals without killing them in a day, right? So you have to titrate it. You have to do a dose response first. You say, 10 PFU, the animals will live for 10 days, and I could see the effect of an antiviral. So that you have to empirically determine the dose that way. Now, they did a challenge study in the UK for SARS-CoV-2 some time ago, and the data are still being analyzed. It's been published already a few times. And they just gave them one dose of virus, right? 10 to the 5. They did, I think it was either TCID 50 or PFU. I don't remember. So to their credit, they did quantify, but they only used one dose. And they gave it to them in a totally artificial manner. They pipetted it into their nose, and then they clipped their nose with a clothespin and made them sit back for some minutes. Right. This is not good. I would like to know how many, and I think with that, 50% of the people got infected. So it was an opportunity to do a dose response, but they didn't. By the way, those people in that study were, if you listen to Clinical Update 1152, Daniel talked about a study where they gave some cognitive tests to these individuals. And it turns out they all have cognitive defects, and they didn't even know it. And many of them didn't even have symptoms. But now they're have problems the rest of their life. And that's why I originally said the study was unethical, because here's a brand new virus. We don't know what it's going to do. They said, they're all young and healthy. They'll be fine. Well, they're not fine. And you learned, and it wasn't fair. I know they agreed to it, but maybe if you had said you could have cognitive defects for the rest of your life. Are you okay with that? I bet a lot of them wouldn't have been okay. So it's a bad idea. Sorry. At what temperature do we incubate the plaque assay? It's typically 37 degrees Celsius, right? Human body temperature. But sometimes you go lower. Sometimes you have mutant viruses that don't replicate well at high temperatures. We go 32 degrees, but mostly 37. Yeah. Yeah. How effective are antivirals against Bell's palsy? No idea. I didn't know they were even tried. Let's look it up, Rob. Antivirals are used to treat, but mostly are effective with steroids. And according to the Mayo Clinic, antivirals added to steroids might benefit some people, but it's not proven. Valcyclovir or acyclovir is sometimes given in combination with prednisone in people with severe facial palsy. So that's an anti-herpes virus, right? I don't know why that would make a difference, but as I said, that it hasn't really been proven. I thought the test line was something sure to be positive. No, the control line is the one sure to be positive. The test line is what you're testing, right? At least that's the way I'm putting it up. One more question. What's the case of Paxlovid being cost effective? Some doctors are hesitant to prescribe it, maybe only for high-risk people. Well, he has talked about it, and he decrees the fact that it can be $1,200 for a prescription, right? He said that's not good, and it has to be fixed. I mean, price should not determine your medical care, but as we all know, it often does, right? I don't know if it saves your life, that's cost effective, right? That's the way I would look at it. If you're high risk, it's worth it. I mean, Daniel's seen many people who haven't taken Paxlovid in time, and as a consequence, they end up passing away. So I think $1,200 is worth your life, right? Jeff asks, can HIV exist outside the body? Its infectivity lasts hours at most outside the body. It's extremely labile virus. So fomites are not going to transmit an infection. Yeah, it's a highly unstable virus. And so, blood and sex are direct ways of injecting the virus into a person. And so they're very effective, obviously, but not fomites, no. Mark says, do you know if Michael Minna found a partner for his low-cost SARS-CoV-2 assay? No, as far as I know, he never did. The $1 dipstick rapid antigen test. I mean, we have rapid antigen tests, but they're not $1, right? So he did not know, as far as I know. Scott says, I once had an instructor who said the only stupid questions are the ones you don't ask. That's absolutely right. That's great. I love that. Let me write that down. The only stupid questions are the ones you don't ask. Only stupid. Excellent. Thank you, Scott. I really like that. Yeah, sorry, the rats are sacked. The mice are sacked to get the semen. They're not going to enjoy it at all. Sorry. Seems like your perspective on rats and nucleic acid tests are based on going back to work, etc. I use gnats for early detection of virus to make informed. Yeah, in the research lab, it's a different story. Yes, most people are interested in when they can go back to whatever it is that they're doing, right? But you say gnats for early detection to make informed decisions. Early detection, yeah, but if you keep detecting it for 2 weeks, 3 weeks, what is the value of that? Especially since you're now over 30 of cycle threshold and there's not going to be any infectious virus there. What is a virologist's favorite way to wash a watermelon? I don't have a favorite way. Just wash it. I don't know. You should. A watermelon is a porous surface. It's going to be very hard to get it clean. What you should do is wash it as long as you can, and then you cut it and you take the skin off, and then eat it. But I'm not an expert on that. You got to ask Michael Schmidt. How's the infectious dose determined? Yeah, so you make dilutions of your virus, and you either put them on cells or give them to animals, and figure out, your readout. What is it? Death or weight loss or fever, whatever it is, the dose that you calculate to do that. Just finished listening to your mini lecture. Does the viral stock initial concentration matter? This will affect the dilutions. Yeah, of course. If you don't know the stock concentration, you're going to have to make more dilutions, right, to cover it. But in many cases, you know how your virus grows, so you can make dilutions that cover it. When you can do -1 to -9, you're going to cover it. Not going to go above that. But you could do fewer, is what I'm saying. Remdesivir is thousands more and logistically tougher. Yeah, IV. But I imagine Daniel might say how much is up to 90% reduction in severe outcomes valued. Exactly. I think that's a good point, John. Thank you. Marburg sounds nasty. Is there any non-symptomatic spread? No, as far as I know, there's no non-symptomatic spread. And in fact, there's no transmission until you get symptoms, right? So non-symptomatic spread. So there's no pre-symptomatic spread. No, only at symptoms. So that's good. So if someone had Marburg and flew somewhere and then got to their place and then got sick, that's good because they wouldn't be infecting people on the plane. They would only be infecting people when they got sick. And that's what happened when an Ebola patient went to Dallas years ago. He flew from Nigeria, went to Dallas, got sick, went to the ER. They didn't know what he had. And by the time they figured it out, people had gotten infected. The nurse got sick, if you remember. John writes, could heat or cold affect vaccination? Sauna, ice bath, localized ice bath, infrared. Not aware of any studies that have addressed this. I mean, perhaps in animals, but I'm not aware of them. Let's see if we can look it up. Well, they've used ice, of course, to reduce pain at the injection site. It doesn't interfere with an antibody response. So that's partially an answer to your question. But I don't know anything about heat. Kang says, is there a direct relationship between infectious dose and R naught? I don't know. I don't think the study has ever been done. So in other words, if you give people more virus, will they transmit it to more people, right? So the R naught is a measure of how many people an infected person will infect. So if you give more virus, do you get more transmission? So in real world, we don't know because it's very hard to tease that study. I have no idea how much virus people are infected with. So you could do it in an animal model. I'm not aware of any that would do that. But in theory, if giving more virus caused higher viral loads, then it could affect transmission. So theoretically, yes, it could be. But as I said, I'm not aware of any study that's looked at that systematically. It might be done. It's a good question, but I don't know of it. Dorothy, thanks for the explanation. I know I will understand plaque assays after I listen a couple of times. Yeah, it will just take a few times. It's fine. And it doesn't matter how many times it takes, as long as you get it, right? Wayne says, are watermelons a major viral vector? No, I don't believe so. But they can be contaminated with bacteria, right? Especially if the farm is using water to wash the melons, to get the dirt off. That's contaminated. So the melons will have bacteria on it. When you slice into it, you will bring the bacteria from the skin to the flesh, right? And you do multiple slices. So it really will depend on the bacterial load, whether it's going to be a problem or not. And then you have the bacteria on the part you eat. So it's probably a good idea to take the skin off so that you don't keep transferring bacteria to you as you eat it. Let's look it up. Yep. They can be contaminated by bacteria in the food chain. It's like listeria. Listeria is often contaminating melons. I haven't eaten a huge amount of melons in my lifetime, but I never got sick from one of them. Doesn't mean anything. You know, cantaloupes in particular, the rough outer skin, that's hard to wash, right? I don't know what you would do. Michael Schmidt, we'll have to ask him tomorrow and TWIM. All right, fun question now. Is there any virus in space? It depends whether there's living things. So we have an International Space Station, right? People on it, they have viruses in them. They have phages that infect their microbiome. A lot of other eukaryotic viruses too. Where else? Well, there's supposedly water on Mars, right? Or there was. So which there might have been bacteria there. And if there were bacteria, there were viruses. Where'd they come from? I don't know. Maybe they evolved there. Maybe life evolved to a certain extent there. Or maybe they were brought from another planet on an asteroid, right? You know, many plants are inhospitable and they can't support bacteria or viruses. They get burned. It's too hot and so forth. But some are okay. Now, there's so many planets out there, right, that it's likely that there's some kind of life on one of them, unicellular microbial life and viruses, but don't know. Most of them we can't get to, right? I mean, the best we do is Mars with these great rovers, and we're learning about that. Nathan says post-viral myalgia, having a very strange and lingering pain in the legs after a mild flu-like thing. Is this the place to ask about this? Well, not really, because I'm not a medical doctor. But you can get post-viral myalgia for sure, and flu for sure can do it. If you want more, you could ask Daniel at microbe.tv. If you send an email and ask him, we'll answer it on his clinical update. Environmental says, I grew 5 gorgeous watermelons in my garden. Cantaloupes were great too. Yeah, so your garden, you can control the contamination. You know, dirt can have listeria. You can keep them off the ground, right? They're growing on vines. Enjoy. Abdul Rahman says that episode with Kate Rubins made me apply for Baylor's space medicine program. That's great. Yeah, Kate is cool. We had her twice. Once on Earth at a TWIM recorded at ASM Microbe. And then she came to us from the International Space Station by a satellite link. And it was great. That was a TWIV. That was really cool. I had to go through a protocol to connect, say all these things. It was so fun. I love that. She's great, to talk to. Scott says, just put the melon in a box with that always warm rock your grandpa found. 5 minutes should kill anything. See, the radioactive rock, right? He says, it's a number I made up. Yeah. There are tardigrades on the moon, so viruses, but will they be active? Well, if there's no host, they're going to be non-infectious because they will degrade eventually without reproducing, right? They don't last forever. You might find nucleic acids, right? I don't know if they've ever brought back, they have brought back moon rocks, of course, but I don't know if they ever ground them up and tried DNA sequencing. Not aware. Let's look it up. Has anyone sequenced moon rocks? Well, apparently they brought back a lot of rocks, and these have been distributed to the research community. But I don't see that anyone is sequencing them. That would be interesting, right? But it'd be really hard to know if it were contamination or not, right? By the astronauts themselves. Huh. Here we go. Visto says, I heard NASA isolated an engineer sneeze still on camera, come back from the moon. Yeah. The people have a lot of viruses and microbes themselves, so that's a problem. John says alien life may be mostly slime instead of intelligence. Well, maybe it's intelligent slime. Brian Cox. Yeah, it could be. There's a lot of slime on Earth, too. Wouldn't make fun of it, but yeah, I understand what you're saying. It might not have progressed as far as we have here on Earth, right? They're all maybe all intermediate stages. Who knows? So environmental says she keeps the melons off the ground with containers. Very good. Brussels sprouts. That's what we do up here. We grow our food. Yeah, I'm sure it's satisfying to do that. That's great. It's Horky says, which methods does one use to determine the lability of viruses? Well, the best way to do it, let's say you want to know how long a virus lasts on a plastic surface. You take a known amount of virus where you've quantified the amount of virus. So you put 1 million PFU on the surface, and at different times you sample it and do a plaque assay and see with time, does the infectivity go away? And it will. And the question is how much time before half of it goes away, something like that. So you essentially subject your virus to whatever condition. You could heat it. You could dry it out. Anything where you wanted to know the effect on infectivity. You could leave it in the sun, right? UV. And then you do a plaque assay to measure the infectivity that's left. Yep. Star Talk had a couple of professors from the University of Florida that attempt to use moon regolith to grow plants. Maybe Rich knows them. Maybe. Maybe. The tardigrades were on a Israeli moonshot that crashed a year or so ago. Oh, they're the tardigrades on the moon. That's where they're from. Huh. Okay, Abdul Rahman is an MD. Post-flu myalgia can be common or severe. If they can get CK measures, it gives a better view. There you go. Mark said, given contamination risks, how are germ-free mice stay uncontaminated? Well, they have very strict conditions for that. And they have sentinel mice that are used to test. So they're germ-free. Right? There are two kinds. They're germ-free and they're specific pathogen-free where they're missing certain pathogens, but not all. But the germ-free, yeah, they don't have bacteria. And it's a very carefully controlled environment. And so the sentinel mice are sampled regularly to make sure that there is no contamination. Yeah. Now, I have never worked with a germ-free mouse. They're kept in isolators and they're born by C-section with germ-free foster mothers. So the isolators have gloves, so you really don't ever take the mice out. They spend their life in there. Jixon used to work on these in Notre Dame. Should ask him next time. Yeah. Oh, look at this. Environmental has indoor hydroponics for growing lettuce over and over. Yeah, it's cool. So we used to have a podcast called, what was it called? I don't even remember. Is it Indoor Farming? Gosh, I can't remember the name of my own podcast. It's defunct, of course. Urban agriculture. And we went to interview the CEO of Gotham Greens. It's on the top floor of Whole Foods in Brooklyn. And they cycle every 21 days. They get a new crop all year round. Amazing. Leafy stuff, but beautiful. Really cool. Oh, I just got rid of the overlay. Thank you. Okay. Vertical farming. That's what Dixon's all about. The indoor farm. And now, of course, it's a whole city that he's interested in. Anybody else have any questions? Folks, it's like 10 of. It's a good time to stop if you'd like, but I'll wait for a question if you also have one. But that was a good session, I think. You guys have good questions, good chat. Hope you got good information. All right. Let's see. There's one more popping up here. What diet would germ-free mice have with no biomes from mouth to bowel to help with nutrient extraction? Sterile. Yeah, I mean, it's not great, right? They're not really that healthy, but they get sterile food, sterilized, autoclaved. And probably special food that provides things that their microbiome would normally make, which they can't. So these are provided in the food. Special. Is there any difference between the intelligence of slime and certain politicians? Probably slime is more intelligent. They react to the environment. They don't do any thinking or scheming or lying and so forth. Louis wants to know the best senior flu vaccine. The one that you can get. Well, seniors should get the double dose, right? Whatever double dose you can get is fine. You can't have flu mist. Over 49 doesn't get flu mist. So it's going to be an inactivated vaccine. Is going to be either grown in eggs or in insect cells. It doesn't matter. But just get the double dose. Well, thank you, Environmental, for chatting. We appreciate your participation. Yeah, you don't have to do other things. It's good to contribute your knowledge. All right, folks, that'll do it for another office hours. I want to thank everybody for coming. 130 folks, 129 likes. I couldn't ask for more. Thanks to the moderators who were here tonight. You want to learn about mycoviruses at some point? Sure. Yeah, we can do that. Maybe get a guest who knows about it, right? So Andrew was here. Barb Mack was here. Vanity Nutrition was here. Tom was here. Les was here as moderators. Thank you so much for moderating. We really appreciate it. Heather, welcome. Yeah, we're just finishing. Watch it later. There's some good stuff here. Really appreciate it. When we back? We are back in 2 weeks. So the next one will be October 16th, Wednesday, October 16th. No office hours next week. October 16th, 8 p.m. Eastern. Come back, bring your questions. In the meantime, I'd like you to be safe. Take care of yourselves, everyone, and thanks for coming. Bye-bye.